Hello, my name's Christopher Bravery. I'm a consulting regulatory scientist at Advanced Biologicals. And today I'm going to talk to you about the quality of plasmids used for viral vector manufacture. I'm going to talk about two situations. I'm going to talk about the plasmid as a drug product or an active substance of a drug product, more as a contrast to the main purpose of the talk, which is to talk about plasmids as starting materials for the manufacture of viral vectors by transient transfection systems. That means I'm going to talk a little bit about quality systems, actually only quite a little bit about the cell banking, and quite a lot about the plasmid specification at the end. But I should say that there are some additional slides in my presentation which I won't show during this live recording, but will be available afterwards, complete with speaker's notes. So I think the first question to ask is the status of the plasmid in the case of viral vector manufacturing. I only raise this because I can't actually find a very clear answer about how the FDA view it. In Europe, the Medicines Directive makes it very clear that plasmids are starting materials. If I use ICH principles, regulatory science, then a starting material is something that's partially or wholly incorporated into the active substance. So on that basis, clearly plasmids are a starting material. Sorry, I tend to repeat this, but GMP is not a grade, it's a quality system. So to call it a grade is a bit meaningless. It tells you nothing about quality. It's a system of for quality assurance more than anything. Although there are, there are important aspects in there, but it doesn't really alter the quality. There isn't per se any legal requirements for plasmids, for starting materials to be GMP. Indeed, a colleague of mine years ago was assessing a dossier for a hormone that was extracted from the urine, which was collected in primary schools in China. So clearly that wasn't GMP, so it can't always be GMP anyway. However, this talk is built on the assumption you're using bacterial cell banks to express, to expand plasmids to use, and therefore the nature of that process suggests to me that it really probably has to be a GMP type system to give you an assurance. But they can only ask for principles of GMP because I can't find a legal basis as such. Which brings us on to quality systems. Now, if you looked at ICH Q7, which you probably wouldn't do for your own local GMP requirements, but you'll see that there's a table in there which gives you an idea of where they think that GMP should start. Now, this was 2010, and in that time in Europe, it showed the same. What it shows is that from the maintenance of the working cell bank forward, which I don't think was best practice even in 2010. Before this particular version of Annex 2 from Europe, which is dated 2018, I think at least 4 or 5 years before that, this table had been altered to look like this. So it starts from the preparation of the master cell bank onwards. So that's when it should be in a GMP environment. And just to show you that this is probably what should be expected, here's a WHO guideline. Now, WHO you can see as like a minimum regulatory system. Indeed, they provide lots of documents for countries that don't have a proper regulatory system to help them set them up. So they've actually now included cell and gene therapy in their table as well, interestingly, from 2016. And as you can see, it's like Europe, it says from the master cell bank. Now, I didn't go looking for FDA because I don't recall ever seeing a table like this, and so it wasn't terribly convenient to try and dig around. But I think the message is from when you prepare the master cell bank, that should be in a GMP environment. I also want to draw your attention to a very nice technical report from the Parenteral Drug Association. Now, this is for excipients, and it walks you through a formalized risk assessment. But excipients are deliberately administered to man, whereas a starting material is never going to be administered to man. So I think the pragmatic approach to quality systems in here may be worth having a look at, because I think you could apply the same principles to your raw materials as well. They don't all have to be GMP for sure, in my opinion. So let's move on to the cell banking system. Now, if any of you have worked in biotech before, this sort of table will be very, very familiar. If you're new to it, and I'm going to assume you might be new to it, I'm just going to walk through why it's laid out like this and so on. The reason I have chosen to use the table from the European Pharmacopoeia is only because it was the smallest, most condensed and compact, simple table that I could find. I couldn't find an equivalent from the FDA, for example. But you'll find similar tables in many other documents, but they're not necessarily focused on plasmids specifically. So I know in the past when I've tried to compare different guidances for mammalian cell lines, you do find some funny inconsistencies between regions, despite the fact that ICH actually has sort of supposedly harmonized that. So I can't swear that other regulators might have slightly different views about what should be done, but they are fairly simple. So I would be surprised if there's anything majorly different to this. What they might do is suggest some slightly different sequence of testing. So when you first get your host cell strain, it's obviously pretty important, as well as making sure you have some knowledge of the provenance of that bacterial strain, that you double check that it is what it says it is on the tin, and that it is free of contaminating bacteria of other sources and bacteriophage, and that that it's healthy, it's viable. Once you're satisfied with that, then you can start your whole process of preparing your plasmids and your plasmid banks in the usual way. All this can happen outside of GMP. And then you will be ready to start bulking up those bacteria into a master cell bank, which you should then do in GMP or principles of GMP. The master cell bank you always thoroughly test. Of course you want to do that. That's the first point. If you've got that wrong, what's the point of moving on to other steps until you've double checked? So now that you will double check again that it's not full of the wrong strain of bacteria, and it's not full of bacteriophage, but importantly now we'll look at genetic stability as well. So you make sure that, see what the copy number is, make sure that the sequence is correct, and so on. And then you would be ready to prepare your first working cell bank. And each time you prepare a new working cell bank, it should be tested once, as per this column. And you'll notice that the column for working cell bank testing is somewhat more limited than the master cell bank. That's because, in part, because the best place to test actually is at the end of production. So that final column means end of production cells. There are a number of other terms for that, so you might see it phrased differently in different guidances. So the idea there is that those end of production cells have not just been through the manufacturing process of the working cell bank, but they've also gone all the way through the manufacturing process of the for the product, or the active substance to be correct. Now, this is quite simple for a plasmid. It's just expanding up the bacteria. But the point is that if you've got contamination brought in from your raw materials for that part of the process, the working cell bank process, etc., then this is the point where you're going to have the best chance of detecting them. And it's also about checking the genetic stability of your bank, making sure it can get to the end of production and still remain stable. Without errors creeping in or it becoming infected with other bacteria. So now for the rest of the talk, pretty much, I'm going to go through the release testing. Now, what I So I obviously went to USP 1047 as primarily a US audience. I thought it would be useful to include the European Pharmacopoeia's 5.14. And I had discovered that the FDA have a very nice guideline for DNA vaccines for IND stage, which I felt contained a bit more information on plasmids than their, say, gene therapy guidelines. So I wanted to draw your attention to that. So I included that in this table as well. First, let's start with the legal basis of these. So USP chapters between 1,000 and 2,000 are general information. That means they're guidelines. The FDA guidance is exactly what it says on the tin. It's guideline. And it's guideline actually for IND phase. The European Pharmacopoeia, in comparison, is a general monograph, which means if you are making a plasmid drug product, you must comply with this monograph. Within that monograph, it describes the use of plasmids as starting materials for viral vector manufacturing and directs you to the same section, pretty much. So, so they are assuming that you would apply similar testing. It's not brilliantly clear. Anyway, I've also slightly, so I based it on the USB table and then added the others in. And I slightly rearranged the order to more align with what I would expect a specification to look like. So usually you would put the appearance at the top of a spec is the first thing you would look at. You just double check that the stuff looks correct. You know, if it's gone brown when it should be clear, then clearly there's something wrong with it. Then an identity test. Now, a test for identity is not quantitative. It's just yes, no. Is the stuff there? Yes, no. So that was all aligned. Restriction digest is a common way of doing it. Then we got purity. And it's actually purity of the DNA forms here. And the supercoiled DNA forms are important for the effectiveness of the plasmid, shall we say. So they're there, and they're in all three texts. USP offers, suggests that this should be above 97%. It's a tiny bit odd to provide a suggested specification of a drug product of this nature, because they should usually be case by case. So I'll come back to that point in a minute. The FDA guidance actually just suggests that a reasonable starting point for IND is at least above 80%. So if you're using a plasmid as a starting material, I would suggest that the FDA's guideline might be a suitable starting point for your own targets. But it's up to you to determine whether that the differences in purity matter to you. And indeed, it might be very easy to get the plasmids at a higher purity than that. So 97 will be great, but it may not be essential, is my point. Now, given USB is supposed to be a drug product specification, it's slightly odd that it doesn't contain total DNA as an item on the specification. All drug products, you need to know how much stuff is in there: total protein, total number of cells, or total DNA in this case. Both the other two texts identify that, and the European Pharmacopoeia includes a couple of different suggested methods. They're not absolutely essential to use those methods, depending on how much DNA you've got in a product. Remembering it might be quite a low concentration for some products, might be a higher concentration for other products. Now let's move on to impurities. So process-related impurities don't normally change much for biotech products between the drug substance, that's the end of manufacturing of the active substance, and the drug product, which is often sent to another site. The concentration's adjusted, it's sterile filtered and filled. So you don't normally retest process-related impurities in a drug product unless there's a particular reason to do that. So it does seem a tiny bit odd to me that USP has included it in what's apparently the drug product spec. It doesn't provide a drug substance spec. The European Pharmacopoeia provides both, so I've put the drug substance section here for impurities because it doesn't list those under the final product. The FDA guideline mentions the same testing. Now, USP also provides some quite specific minimum targets for these impurities. Again, I would argue for any drug product, unless you absolutely know the route of administration, the indication, and the dose that you're likely to administer, and that's always going to be the same, which it never is, then it's a bit dangerous to provide suggested specs. So I don't really think that's a good idea, but they've got some. So it gives you a sort of target thought, where to head for, but don't copy them. Just, consider them if you're in that ballpark, you're probably okay. FDA actually a little bit more aligned maybe with a chemical approach where they often say that certain impurities should be below a certain percentage or ppm. And so they've just sort of suggested keep everything below about 1% and you'll be good to go for an IND. Now, given that we're mostly here talking about plasmids as starting materials, then I think I would err towards the FDA's, try and get it about 1%-ish. But you might even be able to have a bit more than that. I mean, the question becomes, will these impurities have an impact on your manufacturing process? I mean, of course, it's better to err on the side of purity, but if you're having difficulties, I wouldn't lose sleep over it as long as you can show that it doesn't impact the process. Then moving on to the other half of the specification, I've put the term contaminants here, which is the ICH term. Sometimes people tag these as safety. USP had tagged them together with purity. I don't really like this purity, including all the impurities. I prefer to keep those concepts separately. So the purity as in the sense of a purity test, and then have a separate entry that says impurities and lists the impurities. Anyways, but endotoxin has now been removed in the next version of this 1047. It's been changed to be aligned with the other two, actually, which is just that you should justify the level of endotoxin. There are well-established maximum amounts of endotoxin for different routes of administration. So what you normally do is a worst-case calculation based on a worst-case body weight and the volume that you're going to administer, the maximum volume you're going to administer, and then calculate what's the maximum endotoxin you can have in your drug product, and preferably try and keep a bit below that as well if you can. So that's really what the other two texts describe. The FDA provides a bulk, a drug substance limit, if you like, a suggested limit of around 40 or less. And then sterility is straightforward because it should be negative. And now we get to potency. Now, the FDA and the European Pharmacopoeia both make it clear that a potency assay is a bioassay and that it should measure the biological activity of the gene product of the plasmid, because that's would be the potency for a drug product. And they also maybe remind you about the need for reference materials, because a potency assay needs a reference so that it can be measured in units of potency. So then when we look at 1047, it's a bit confusing. The first thing is it includes absorbance A260, which is the total DNA content test that I said was missing. So why is it there under potency? And curious that it hasn't been picked out separately. It's just jumbled in as a possible method for potency. What, total content? Very strange. And then the other two methodologies listed in 1047 are ELISA and RT-PCR, both of which are physicochemical methods. So they cannot measure potency. A potency can only be measured by a bioassay. They might be used as the readout of a bioassay. So a bit odd. And when I looked at the text, I couldn't find anything particularly on the plasmid area that talked about the need for a potency assay. Now, when it comes to using a plasmid as a starting material, I recommend you don't call it potency. You should call it biological activity. And then I think the question becomes, do you develop an assay that confirms that each batch of plasmid can produce the gene product, and that gene product is active? Or do you produce a scaled down model of your process so that you can also evaluate the transfection aspect for each new batch of plasmid? Or indeed, is there a difference between those two, except the label you might put on it? But clearly, from a manufacturing point of view, that's what you're interested in. You're looking at this as a material. You've hopefully established the critical materials attributes, which is a way of saying quality attribute for raw materials and starting materials, rather than calling them quality attributes. Because you do want to check each batch, probably. But maybe you'll become confident enough that it's not always essential. But again, that's for you to determine. And then quickly, finally, because I feel like I'm probably up to time, there's some general tests. Now, these general tests may or may not be relevant for a starting material, but for a drug product, one really important general test, together with the content of each drug product container, is that you can extract that content from the container. And indeed, the test is harmonized between USP and the European Pharmacopoeia and JP. So it's slightly odd that USP forgot to put it on, given this is a drug product spec, because it's a very important test, as is determining that your whole batch is consistent from beginning to end. And there's a monograph for that too. Also, if you've lyophilized, which obviously some people do with plasmids, then residual moisture is actually pretty important. pH, probably important, but actually whatever the pH is, you've determined through your formulation studies, so I'm not sure why you would put a suggestive range in, really. You might want other tests like osmolarity or something, depending on how you're using it. And that's it. So plasmids are fairly straightforward. Probably I made it sound more straightforward than it is in reality. But as materials go, these are quite straightforward. And they should be made under a quality system. And I think principles of GMP probably is the right approach if you're using bacterial banks. If you were chemically synthesizing them, I think this would be a different discussion. I don't have time to discuss that right now. You will need to provide all these details, including the fact you should have a reference material for your plasmids as well. And I've included some additional slides at the end of this presentation that you can review for yourself, including speaker's notes, which are excerpts from the European Public Assessment Reports and BLA documents to give you a feel for what the agencies have actually asked for. And my final comment is just remember the specifications are your responsibility and you need to determine what the appropriate acceptance criteria are. And on that note, thank you very much. And I will take questions later.